All right, so the first step in Krebs's work was to make the connection here between oxaloacetate and citrate. He's working with a student of his, I believe, a guy named Johnson, and what they showed was that you can take oxaloacetate and make it into citrate. They found a way to do that chemically, which meant that if you could do that, then this whole thing would become a cycle, would just become this closed loop. Because then, if you made citrate, isocitrate, and all these other things, you just make the oxaloacetate, which could then make the citrate again. But the question was this. Notice citrate has 6 carbons. 1, 2, 3, 4, 5, 6. But oxaloacetate only has 4. 1, 2, 3, 4. So the question then became, where did those extra 2 carbons come from? And in their experiments, they didn't necessarily know where those 2 carbons would have come from in the actual cells themselves. And so this was the key step, because remember, what we're trying to explain is how sugar metabolism uses carbon dioxide and oxygen and how that's related to these organic acids that you see here. What Krebs then suggested was, okay, look at the end of glycolysis. We take sugar, feed it into glycolysis, and we make pyruvate. What if those 2 carbons are coming from pyruvate? In fact, in order to get those 2 carbons, now remember, pyruvate has 3 carbons, but in order to get those 2 carbons, we'd have to remove one of the carbons on pyruvate. And notice there's already a carboxyl group on that pyruvic acid. If we remove the carboxyl group and release it as carbon dioxide, then we can take this group, which is acetate, these remaining 2 carbons would be an acetate group, which could then supply the 2 extra carbons here on citrate, and that would close the loop and connect it to sugar metabolism. Not only that, but it would also explain where the carbon goes. Because think about this, if we're going to completely oxidize glucose, we take 6 carbons and we have to make 6 carbon dioxides. So can we make 6 carbon dioxides here? Yes, if you look here, this pyruvate has 3 carbons on it, 1 comes off here, 1 comes off here, and the other comes off here. So those 3 carbons come off as carbon dioxide. There are 2 pyruvates per glucose, therefore a total of 6 carbon dioxides per glucose, and this would completely oxidize the glucose. So Krebs' suggestion here took a long time to actually show us what was going on, but it made perfect sense with what they understood, and it connected it completely with oxidation of glucose, except for one thing. And this should be the thing that keeps in your mind. We don't know where it is that the oxygen is used, because nowhere in this scheme is oxygen used, only carbon dioxide is given off. So that was the hypothesis. Now what they need to do is they actually have to test it to see if the hypothesis is correct. So the first thing, well, maybe not the first thing, but one of the key results, early key results that Krebs obtained, he obtained with another guy named Eggleston, and a few years later after he made the hypothesis. What they did is they showed in a beautiful experiment that if you feed cells pyruvate, you get carbon dioxide off as if the pyruvate is completely and utterly oxidized. And not only that, but you use up the oxygen. Now remember, if we're going to connect this to carbon dioxide, we'd have to show that the pyruvate is producing the right amount of CO2. There's nothing else going on. It's not used for some other reason. And it should somehow use oxygen, even though we can't see it in this scheme. So here's what they did. They fed cells a bunch of pyruvate, and they measured the oxygen that was being consumed, and they measured the carbon dioxide that's being released in machines like this one. Okay, it's a relatively easy thing to do. Now let's look at this equation. If we're going to take the pyruvate and completely oxidize it, which is what we think this is doing, we think this is completely oxidizing the pyruvate. If that's true, then the balanced equation would be this. C3H4O3 would make 3 carbon dioxides and 2 waters. Again, to make the 2 waters, I have to balance the hydrogens. 3 carbon dioxides have to come off, but then to balance all the oxygens, which there'd be 8 oxygens over here, I'd have to have 8 oxygens here. So to do that, I'd have to feed in 2.5 oxygen molecules. Now this may look weird to you, but realize these are really moles of each thing, not individual atoms or molecules. So this is 2.5 moles, or if you wish, 2.5 O2 is just an oxygen. But anyway, if you look at this, this is 2.5 O2, so that's a total of 5 oxygen, plus 3, there's 8. So this would balance. So what that means then is if you feed in pyruvate and the pyruvate is completely oxidized, we should release 2.5 moles, or sorry, should consume 2.5 moles of oxygen and release 3 moles of carbon dioxide. So that's exactly what they expect. They expect 2.5 moles of oxygen should be consumed for every pyruvate, mole of pyruvate that's added because of the balance, and 3 carbon dioxide should be released for every mole of pyruvate added. That's the prediction, and here's what they got. In their actual observed results, they got 2.54 moles of oxygen consumed per mole of pyruvate, and 3.01 moles of carbon dioxide per mole of pyruvate, right? And now that means what? Well, you tell me. Does this observation support, contradict, or say nothing about Krebs' hypothesis? Okay, well, if you look at this, notice the predicted and the expected results, and the observed results are nearly identical. Not exactly the same. Why not exactly the same? In fact, this is slightly more than that, and this is slightly more than that. Well, remember, every measurement has error associated with it. If anybody says that my measurement is perfect, they're lying or they're misinformed. You cannot make a perfect measurement. So the results that you obtain here are going to be sloppy. There's going to be a little bit of slop in them. And notice how close this is. It's to 2 decimal places. We're dead on. Or at least to 1 decimal place dead on, and 2 decimal places if you round. So this is as close an experimental result as you're ever going to get. This is beautiful. So the expected and observed results are essentially identical. And that is expected under the hypothesis that the pyruvate is completely oxidized, which it would be if this hypothesis were true. Therefore, these experimental results support the Krebs and Johnson hypothesis. Do they prove it? Of course not. Of course they don't prove it. Remember, we're not in the business of proof here. In science, all we ever ask is this system logical, and does the experimental results support, contradict, or say nothing about it? That's all we can do. This is supporting, but not proving, that this is a potential thing that's going on. Okay, it's consistent with it is another way of saying it. But we need more. We need more experiments than just that. Well, there's another thing that was discovered about this time, or actually I think it was discovered before this, is that malonate, there's a type of molecule, it's a drug called malonate. And one of the things that's known about malonate is that it inhibits oxidative metabolism. You give cells malonate, and they stop consuming oxygen, and they stop producing CO2. And if you give enough malonate to a human being, they'll die because they won't be able to complete their oxidative metabolism. And we know now that it's because they run out of ATP. Remember, the whole purpose of this is to make ATP. So we know that malonate inhibits oxidative metabolism. But what was discovered is that malonate does so by inhibiting this enzyme right here, succinate dehydrogenase, that blocks this step of taking succinate and making it into fumarate. Now, does that observation support, contradict, or say nothing about the Krebs and Johnson hypothesis? Well, again, there's no oxygen that's here, but malonate stops individuals from being able to use oxygen. So whatever connection there is in this cycle, it must be somehow associated with the oxygen, because malonate blocks this part of the cycle and it blocks oxidative metabolism. So therefore, it supports it. It's showing that this particular scheme is somehow related to oxygen use as well as CO2 production, because when you have a poison that blocks part of it, it blocks those two things. So that supports it. Again, does it prove? No, of course not. But what else is there? Well, there's more evidence. In fact, this observation led Krebs and others to start seeing what would happen if you started putting in malonate into cells. Now, let's do that. Let's make a prediction. If this is true, now again, we're not saying that this is true, but if this were true, pyruvate comes in here and the whole cycle is being driven by pyruvate being put in, what would happen to pyruvate consumption? Now, this is just talking about oxidative metabolism, not consumption of pyruvate. But if this is what's happening, what would you expect to happen to the pyruvate consumption if you put malonate into cells? And what would you expect to happen if you put in excess fumarate, if you put in more fumarate? And what if you put in both at the same time? You put in excess fumarate and you put in excess malonate, or some malonate. Okay, so let's make those predictions. Let's test this out. First of all, What would happen if you just put in fumarate without any poison? What should happen to the rate of consumption of pyruvate? Okay, well, you should, since you're increasing the amount of fumarate, cause this reaction to increase in its rate, so you'll make more malonate, which means you'll make more oxaloacetate. Everybody see that? The fumarate, since you're increasing the amount of fumarate that's there, that reaction has to increase, and so you'll make more malonate. If you make more malonate, then by the same principle, you have to make more oxaloacetate. If you make more oxaloacetate, you're going to make more citrate, which means you need more pyruvate, which means the pyruvate oxidation will have to increase. The rate at which your pyruvate consumption will occur has to increase. But notice, let's complete the loop. We make more citrate. Therefore, we make more isocitrate, which makes more alpha-ketoglutarate, which makes more succinate, succinyl coenzyme A, which makes more succinate, and then we make more fumarate again. So look at this. Fumarate will have to act as a catalyst. If this hypothesis is correct, the fumarate should act as a catalyst. And it was shown actually in another paper that that is true. Fumarate does act as a catalyst. And it's going to go do something. This is what we predict. Okay, now what would happen if you don't put in fumarate, but you do put in malonate? Well, if you put in malonate, it blocks this step here. Now, I have a certain amount of fumarate already in the cells. I'm not adding it. I don't have to add it. It's already there. But the cells then will make the fumarate into malonate, make it into oxaloacetate, use pyruvate to make it into citrate, and then isocitrate, alpha-ketoglutarate, succinyl coenzyme A, succinate, and it'll stop there. So eventually we're going to use up the fumarate. If we use up the fumarate, we can't make malonate. If we can't make malonate, we can't make oxaloacetate, which means we can't make citrate, which means we can't increase the consumption of pyruvate. We can't use pyruvate. So putting in excess fumarate will increase the consumption of pyruvate, if this is true, and putting in malonate will decrease it, stop it eventually. But what if we put in both? Well, if we put in both, we're going to have the malonate in here, and it'll stop the whole cycle here, so succinate will build up. But if I put in fumarate, that should rescue it. The amount of fumarate that I put in should actually make the pyruvate consumption continue, because I'm adding fumarate as if this were operating. And so therefore, the more fumarate I add, the more pyruvate consumption I should get, even if I'm adding malonate. All right? So those are our predictions. Put in fumarate, I expect pyruvate consumption to go up. Put in malonate, I expect it to go down and eventually disappear. Put in both fumarate and malonate, I should get more pyruvate consumption than I did if I just have malonate in there, and the amount of pyruvate consumption I get should be proportional to the amount of fumarate that I add. Okay? Now, make sure all of those statements make sense to you. Make sure you follow the logic for all of those statements, because here are the data. These are the actual data. Now, here's the control. And what they're measuring is the consumption of pyruvate in this experiment. In the control, they added nothing. They just put the cells into the chamber and measured the amount of pyruvate that's being consumed. And that's the baseline measure that they got. Okay, now remember, what do we expect if we add fumarate? By the argument that we just made, we expect more pyruvate consumption to occur. Here's where you add fumarate. That F means I'm adding fumarate, and look what happened to pyruvate consumption. Okay, it went up. Now, what if we just add malonate? That's the poison. That's what the P stands for. Well, we expected that to go down, eventually stop, because the fumarate eventually run out, and it can't make any more because I'm blocking fumarate production from succinate. And this is now the pyruvate consumption. It is way smaller than the others. But now what if I add both? Well, if I add both, I should rescue this. I should be able to recover this. And the more fumarate I add, the more I should be able to rescue it. Okay? Well, that's what this part of the experiment is. Here's where they added the malonate, the poison, plus fumarate at a low level. Here's where they added the malonate plus fumarate at a medium level. And here's where they added malonate plus fumarate at a high level. So these three are this. The malonate concentration is the same in each of these three. All they did was increase the amount of fumarate each time. Low amount of fumarate, medium amount of fumarate, high amount of fumarate. And this is the result they got. It's linear. So it is exactly what we predict. Everything that was predicted here is exactly what we see in these results. The malonate inhibits the production of or consumption of pyruvate. Adding in fumarate increases it, rescues it, and that is completely consistent with this being a cycle that uses pyruvate, that is taking the pyruvate in, that's coming from glucose, ultimately through glycolysis. So this connects carbon dioxide production with sugar metabolism. But we're still missing, somehow we are still missing the connection with oxygen. We know oxygen is somehow involved here from the other experiments, but whatever's going on, this is where the CO2 appears to be coming from. So now we have two open questions. One, where is it that the oxygen is being used? This hypothesis does not explain that. And the other open question, even though it says first open question, is how is it the pyruvate is fed into the cycle? So Johnson and Krebs really just said it gets in here somehow, and they didn't have all the details down. So it turns out that question was really difficult to answer. It took a fair amount of effort to figure out exactly what was going on. Again, you can sort of predict that that carboxyl group is going to come off as a carbon dioxide, but exactly what else is going on here wasn't obvious. What was discovered by Fritz Lippmann, among others, was that coenzyme A, that same coenzyme A that we saw in the succinyl coenzyme A, is involved in taking the whatever product from pyruvate is and feeding it into that reaction and leading to from oxaloacetate to citrate. And they elucidated the role of it in pyruvate oxidation along with others. Krebs, for example, also credited these two, Severo Ochoa and Feodor Leinen, that they established that the coenzyme A itself is the intermediate that really reacts and pulls this into the Krebs cycle. But what's happening exactly in this situation? Well, we already know that we're expecting this carboxyl group to come off, and what Lippmann and the others showed was that it does. The carboxyl group does come off. And what's left over is this, which is an acetate group, acetic acid, which is essentially vinegar, or the conjugate base of vinegar. What they showed too was that the coenzyme A replaces this carboxyl group here through a sulfur bridge, and we end up then with this molecule, acetyl coenzyme A, and acetyl coenzyme A then is what is donating the acetate group, the two carbons, onto the oxaloacetate to make the citrate. But they also showed that this reaction is a large, very complex reaction controlled by one enzyme complex, and it is thermodynamically favorable from pyruvate to oxaloacetate. That's an oxidation step, and so it releases a lot of free energy, which the cell then can capture in the form of NADH by reducing NAD. So this is the step. This is the critical step between glycolysis that we saw earlier and the Krebs cycle. So here's the modern understanding of what's going on in most cells that use oxygen. The pyruvate is produced by glycolysis. Glycolysis, by the way, occurs in essentially all cells we've ever looked at, and it occurs in their cytosol. That pyruvate then is produced in the cytosol. And then the question was, well, where do all these other things occur? Turns out the rest of them occur in the mitochondrion. So the pyruvate has to get into the mitochondria. We'll get to that in a moment, but the pyruvate then goes through this reaction to make acetyl coenzyme A. The acetyl coenzyme A is then fed into the Krebs cycle, and this is just your book's rendition of the Krebs cycle. It has all the stuff that we talked about before. So this is the A level of the Krebs cycle. The B level is the same thing, but without the structures and the enzyme names. To get the enzyme names again, of course, just go up to here, and each one of these will tell you what the enzyme is. But then that is our modern understanding of the Krebs cycle. But like I said, this all occurs in the mitochondria, and all the enzymes that drive this series of reactions are occurring in this mitochondria. So that was even more difficult. We had to figure out, okay, well, what's going on with the pyruvate? How does that get into this mitochondria? Well, what would you predict? What kind of transport? We've already studied this in previous lectures. What sorts of transports do we know? We know 4 of them. We know that there's simple diffusion. We know that there's diffusion through ion channels. We know there's facilitated diffusion, and we know there's active transport. So which of these would you predict would be used? Well, we can eliminate simple diffusion because you can see right here that the pyruvate, actually go back up here, the pyruvate has a charge. So it's not going to go directly to the membrane. So simple diffusion is out. Channel, no, it's not an ion. I mean, it is an ion, but it's not really. It's way too big for an ion channel. So that leaves us then with facilitated diffusion or active transport. And the question is, are you trying to absorb the pyruvate and get essentially all the pyruvate out of the cytosol, or are you happy with diffusion where it'll equilibrate? Well, this whole process requires all of the pyruvate to get into the mitochondria. And so we would predict that whatever's happening here, we expect it to be active transport. And that is correct, but also there is kind of a facilitated diffusion that's going on as well. So here's a summary of what was discovered. And in fact, this was worked out only very recently. The details of all of this were worked out only in 2012. But glucose goes through glycolysis, makes 2-pyruvate. That occurs in the cytosol. In the process, it produces 2-ATP and 2-NADH. The pyruvate then has to get through 2 membranes of the mitochondrion and into this space right here. So here's how it functions. This right here is an active transporter. It literally actively transports all of the pyruvate in the space between the 2 membranes out between that space. So it's constantly pumping pyruvate out of here, which means that there's always pyruvate in the cytosol because that's produced by glycolysis, and there's a concentration gradient across this outer membrane. So this portion uses something like facilitated diffusion. The pyruvate just diffuses through, but then this active transporter pumps it out of here, and so it's constantly diffusing in and constantly being pumped into this portion of the mitochondria where the enzymes of glycolysis can work on it. But once the pyruvate is in here, this active transport is using ATP. So we can think of it as using these ATP. So by the time you get the pyruvate inside the mitochondrial inner membrane, this ATP is gone. And the only thing you have from glycolysis are the 2 NADHs. Right? So once you've got that, then we go through the Krebs cycle inside of here, along with pyruvate oxidation. So we oxidize the pyruvate here to make the 2-acetyl coenzyme A. That occurs also inside the mitochondrion. And then we can run through this whole process. Great, so that question is now answered. What we do not know still yet in our lectures is how the oxygen is used. We do know, as scientists, we actually, that was discovered after the Krebs and Lippmann had done their work, but basically we still don't know where the oxygen is based on what we've studied so far. So just to finish this off, these 2 individuals, there's Hans Krebs and here's Lippmann. These 2 gentlemen won a joint Nobel Prize in physiology or medicine in 1953. And the first one, of course, was for the discovery of the citric acid cycle, which we now today oftentimes call the Krebs cycle in his honor. And Lippmann won for his figuring out what was going on with coenzyme A and its importance in feeding in glycolysis into the Krebs cycle.